There we go. So without further ado, we'll start with our trainee speaker, Donovan McDonald. Donovan is a PhD candidate at the University of Western Ontario, and today he'll tell us about the molecular consequences of mistranslating tRNA serine variants. So take it away, Donovan.
Awesome. Thanks a lot, Matt. All right. So thank you for the kind introduction, Matt. My name is Donovan. I'm a PhD candidate in Martin Dunwall's lab at Western University. And Happy New Year's, everyone. I'm really excited to share my work today. Just get this going. All right, so before I begin, I want to start by, sorry. Okay, I want to start by acknowledging all the individuals that have helped make this work possible. So first of all, my lab, Martin Dunwall, my supervisor, and my many lab mates over the years. I've highlighted today two undergrads who've directly contributed to this work. That's Christopher DeLuca and Rebecca Dibb. I also want to thank collaborators, Patrick Lajoie and Dr. O'Donoghue at Western, and especially Matt and Chris Brandel, whose work have really set the stage so that I could do some of the work that I've been doing. So thank you. So my work revolves around these molecules, these tRNA molecules. These are the master decoders of the cell. And they decode mRNA blueprints into functional proteins. And they do this through the pairing of the anticodon sequence with the cognate amino acid that gets amino acylated to the tRNA. And this amino acyl tRNA then participates in translation at the ribosome. Mutations in the anticodon sequence of the tRNA can disrupt this pairing and can yield tRNAs that still get charged with the same amino acid, but bind to the wrong codon. And this can cause inaccurate protein biosynthesis or mistranslation. Now, Matt spent a lot of time in his PhD and beyond studying these tRNA variants, and he actually found that for a long time, we've been underestimating the frequency of mistranslating tRNA alleles, some of which are as frequent as in 8.5% of the human population, which is a pretty high number for such a drastic change in function of such a crucial molecule. And so this really builds a strong rationale to try and understand exactly how these tRNA variants may be contributing to various processes like disease and aging. So mistranslating tRNA variants have been studied in various model systems to date. These include yeast, cultured mammalian cells, and even fruit flies. And the common denominator in all of these models is that mistranslating tRNA variants can cause detrimental effects to these cells. Now, despite all of this good work, we still don't quite have a good grasp on exactly how these mistranslating tRNA variants induce these negative effects on the cell. So that's really what my focus has been throughout my PhD. So to try and study this, I have used a genetic model that's been pioneered by Matt, where we express serine tRNA variants in yeast cells. Today, I'm going to talk about tS, mistranslating tRNA variants. And there are two of them that I use in this study. The first is tS weak, which causes 0.5% mistranslation, and tS strong, which causes 5% mistranslation. And these both cause proline to serine misincorporation in the cell. Now, when we express these mistranslating tRNA variants in yeast cells, what we find is that the tS strong allele actually causes a growth defect. So here's a typical growth assay. This is the common way in our lab that we track growth defects in yeast cells. And each row corresponds to a different sample. So in descending order, we have control cells, cells expressing a wild type serine tRNA, cells expressing tS weak, and then cells expressing tS strong. And what we do is we spot single droplets of 5-fold dilutions of these samples onto an agar plate. So from left to right, we have serial 5-fold dilution. And if we focus on this, the most dilute column on the far right here, you can see that tS strong has fewer cells than the control cell. So this is indicative of a growth defect. Now, our lab is very interested and has a strong expertise in protein quality control. So we really wanted to tackle exactly whether or not these mistranslating tRNA variants might be impairing protein quality control. And our original hypothesis was that these mistranslating tRNAs yield the production of improper proteins that misfold and accumulate in the cytosol, and this would be toxic to the cell. Now, we've tested this through numerous different assays thus far. I'm just going to show one for the sake of time. And this is the HSP104-YFP aggregation assay. And the way this works is that HSP104-YFP forms fluorescent foci when aggregates are present in the yeast cells. So we just take a look on the far left panel here. These are control cells, and you can see the HSP104-YFP has more or less a diffuse signal throughout the entire cell. This is expected in cells that don't have any aggregates, like these control cells. Now, if we take a look at the P2S strong cells, you might the expression pattern is very similar to the control cells, right? So our initial indication is that there is not an overwhelming abundance of protein aggregates in these cells. So next, what we did was we stressed the cells further. We wanted to, first of all, check if our assay is working properly. And second of all, we wanted to test whether or not a second stress can reveal a phenotype in these P2S strong cells. So if we just focus again on this leftmost panel in these cells that we heat shocked at 42 degrees Celsius for 1 hour, you can see HSP104 is forming these fluorescent foci, which are indicative of protein aggregation. And if we take a look at these P2S strong cells, again, we see these foci, but there's not a major difference from control to P2S strong. And this is nicely quantified here, showing there's no difference between P2S strong and control cells when we count the amount of HSP104-YFP foci per cell. Now, these findings and also the findings of several genetic and biochemical assays have indicated to us that there's no real evidence suggesting that P2S strong induces a massive protein misfolding and aggregation in the cells. And so we had to reject our initial hypothesis, and it was, it took us a while to sort of figure out exactly what was going on in these cells. But eventually we happened upon nascent polypeptide quality control. So today I'm just going to describe one pathway in nascent polypeptide nascent polypeptide degradation. This pathway is involved in degradation of nascent polypeptides under permissive conditions. So this is not stress-induced. This is just general maintenance of nascent polypeptides. What we have is ZUO1, which is a J protein, SSZ1 and SSB1R2, which are all HSP70s, and the E3 ubiquitin ligase, LTN1. They all cooperate to facilitate polyubiquitination of nascent polypeptide chains. Now, I was very interested in this pathway because I actually found that ablation of these chaperones interacted genetically with P2S strong. So first of all, I just want to remind you at the top here that in wild type cells, that the expression of P2S strong induces a growth defect when compared to control cells. Now, when we ablate SSZ1 expression in these cells, you can see that this growth defect is actually lost in these cells. So ablation of SSZ1 seems to rescue the growth defect associated with P2S strong. Ablation of ZUO1 behaves very similarly to wild type cells. However, ablation of SSB1, SSB2, and LTN1, which are all, again, these are all members of this pathway for nascent polypeptide degradation, they all rescue the growth defect associated with P2S strong. So this is very strong genetic evidence that strongly suggests that this pathway is in some way, shape, or form involved in the growth defect associated with P2S strong. So we know that these proteins are all involved in degradation of nascent polypeptide chain. And so we wanted to track directly what's happening in these cells, what's happening to the nascent polypeptides in these cells. And to do so, I treated cells with puromycin. Puromycin is an amino acyl tRNA analog which gets incorporated into the growing polypeptide strand, and then immediately these polypeptides will be ejected into the cytosol. Now, what's crucial about puromycin is that it is immunogenic. So we can actually visualize these nascent polypeptides using an antibody. So that's what I did. I treated these cells for 15 minutes with high concentration puromycin. I lysed the cells and then performed an immunoblot. What I found was in cells expressing p-tau strong, we see an overabundance of these nascent polypeptide chains compared to the control cells. That's quantified here on the right. Now, something else that was very interesting was if we chased away the puromycin for 5 minutes and then lysed the cells, what we found was there was a very rapid degradation of these nascent polypeptides. So this exhibits the tight temporal control of these nascent polypeptides, and we believe this indicates the importance of this regulation to the cell. Now, we showed that p-tau strong cells accumulate nascent polypeptide chains, but we weren't quite satisfied yet. We wanted to assess whether or not this accumulation was due to impaired ubiquitination of these nascent chains, or are these chains just being, is the cell failing to clear these nascent polypeptides? So to this end, we performed immunoprecipitation of these puromycin-labeled nascent polypeptide chains. Now this figure is very complicated, but I tried to highlight the comparison that I thought was most interesting, which basically is showing that cells that express p-tau strong, here when we immunoprecipitate puromycin and blot for ubiquitin, we see that the ubiquitination status of these puromycin-labeled nascent polypeptides is much higher in these p-tau strong cells compared to control cells on the left. That's quantified here on the right. So in summary, I showed you today that p-tau strong induces a growth defect in yeast cells by impairing the clearance of these polyubiquitinated nascent polypeptide chains. And so as a result, these nascent polypeptide chains accumulate in the cell and perhaps cause toxicity. So today I just talked about P to S mistranslation, but we also study different types of mistranslation, and interestingly enough, these different types of mistranslation cause their toxicity through distinct mechanisms, and I'll be happy to elaborate on that in the Q&A session. But for now, that's all I have for you. Thank you so much for your time and attention, and if you have any questions, I'm more than happy to answer them.
Awesome. That was a great talk. Thank you, Donovan. I was really excited about that work. Do we have any questions? Feel free to either raise your hand and we can unmute you, or you can type them in the chat and I can read them out. Michael, I can unmute you. Go ahead.
So I'm trying to get ready to go to lecture here, so I was listening with one ear, but I'm finding that your puromycin stuff is really quite interesting, and I may very well reach out to you. We've done a lot of puromycin incorporation assays ourselves. We haven't done the chase aspect, which is novel to me, but the one thing that I was going to mention is that we use a PDR5 knockout background to allow the puromycin to enter the cell more easily. So if that's something that you're interested in following up with us, we're happy to kind of chat with you some more about that, because I think you've got some really kind of cool stuff going on here with the puromycin incorporation assay, and we might be able to contribute to it. So maybe we can talk at some other point.
Yeah, sure. Thank you.
Walid, go ahead.
Yeah, really nice talk, Donovan. Can you comment on the HSP1/O4 levels not being affected in this scenario? That's surprising in a way.
So, sorry, this. So, are you asking if the expression level changes?
Yeah, exactly.
So this is actually an ectopic, this HSP104 allele is expressed on an ectopic plasmid that has a constitutive promoter. So while native HSP104 does go up in these p-strong cells, this HSP104 YFP should be stable. And
Oh,
I
it
have
was not
plotted this and
endogenously tagged. Okay, I see.
yeah, so
Okay,
I
I
have plotted
misunderstood.
this and they are stable, the expression.
I see. Okay.
As a kind of follow-up on Walid's point, Donovan, I haven't shared this with you, but I have proteomic data from a bunch of alanine misincorporating strains, and HSP104 does go up quite a bit in the strains. It's the highest, the protein that goes up the most. It's about 4-fold increase. So
Oh, wow.
even though it's not having an effect, we do see it go up.
Yeah, it goes up in these cells too. Not well, not so drastically, maybe a 50% increase at the protein level. That's what we see via Western blot.
Yeah, makes sense. Awesome. I think to stay on time then, we're going to move on. Thanks, Donovan, for that really exciting talk.
Thank you.
Okay, so next we have Dr. Walid Houry, who is from the University of Toronto. And today he'll tell us about the development of novel anti-cancer compounds targeting mitochondrial protein homeostasis. So Walid, go ahead and share your screen. Looks good, but you're still muted.
Okay, sorry, I was muted. Everyone can see my screen. Okay, excellent. So let me just open the pointer. Okay, thanks very much, Matt, for the introduction. Also, I want to thank Kathy and Fabian from the CSMB committee and the rest of the CSMB committee for organizing this very nice seminar series. So my lab is working on understanding protein homeostasis in the cells. So to this end, we've been working on several chaperones. We discovered a new chaperone complex, but also work on proteolytic complexes. And we do a lot of the biochemistry and the cell biology to understand mechanisms of function. At the same time, more recently, we became interested in targeting these chaperones and proteases to develop new antibiotics as well as anti-cancers. So this is a fairly new undertaking in the lab, and I want to discuss with you today our recent efforts on developing new anti-cancers targeting a mitochondrial protease. So this work is the hard work of several people in the group, especially Mark Mabangalo, who recently left, Keith Wong, who's still here, Mariam, Elisa, Foning, and Emily. We've been collaborating very heavily with several people in the Department of Chemistry at the University of Toronto, especially Rob Beatty, who's been a long-time collaborator on the medicinal chemistry effort. And we have other collaborations, including Anne-Claude Gingeras, Louis Kay, and more recently, Ed and Lee on new types of anti-cancers. So the protease that I'm talking about is really present in the mitochondria in mammalian cells. It's called the ClipXP complex. It consists of a cap as well as a core. The cap is called the ClipX and the core is called the ClipP. It's present in the mitochondrial matrix, in which you find other chaperones and proteases like the HSP60 chaperone, the TRAP1, which is the HSP90 equivalent in the mitochondria. And there are other proteases like LON, which is another major protease in the mitochondrial matrix. So the ClipP itself consists of 14 subunits. It's a 14-mer, and it forms this nice cylindrical type structure. Each protomer looks something like this, whereby you have what we call the handle region and a core or a head region, and then there are these axial loops. So 7 of these come together to form one ring, and then another 7 form another ring, and then the 2 rings come together to form the cylindrical structure, which is a 14-mer. And proteins have to enter this cylinder through an axial pore, which is shown here in a top view. The targets for the ClipP in the mitochondria are known through mainly proteomic studies, but actually detailed biochemical studies have not been done. This is something we're working on. The ClipP is also present in bacteria, and much more is known about the ClipP in the bacterial system. In terms of the mitochondria, we know that it targets several of the Krebs cycle enzymes, as well as several of the respiratory complexes and many of the mitochondrial ribosomal proteins. The interest in ClpP became much more once it was found that it was upregulated in cancer. And here I'm showing you just an example of different cancers and the quantification of the ClpP in these different cancers. So if you just concentrate, let's say, on this Western blot here, so this is a human prostate tissue and showing the levels of the ClpP in the tumor versus the levels of the ClpP in the normal tissue. And you can clearly see that it's very highly upregulated. And this has been found in many different cancers. And that's why many groups from the clinical aspect as well as the medicinal biochemical aspect started investigating the mitochondrial ClpP. So ClpP works, as I mentioned, with a cap ClpX, and the structure looks something like this, whereby you have the cap, which is a hexameric ClpX, coming on top of the ClpP tetradecamer, and then you have a protein that is being captured by the cap, unfolded, pushed through and into the ClpP cylinder where degradation takes place. And this is a schematic of the mechanism whereby you have a folded protein that's physically unfolded by the cap in an ATP-dependent manner. And then this unfolded protein is translocated into the green core, the ClpP core, where it's degraded. So ClpP is a serine protease. You have 14, because I said it's 14, it's a 14-mer, so you have 14 Sear-His-Asp catalytic triads within the chamber, and therefore these 14 catalytic triads are degrading or chewing up the protein into small peptides. And typically you get peptides that have around 10 residue sizes. It's, of course, a Gaussian distribution of sizes, but the average is around 10 residues. So it's a really very processive protease whereby you give it something that's 1,000 residues, you only get 10 residue peptides. You give it something that's 100 residues, you get back 10 residue peptides. So for my presentation today, I'll discuss our efforts at developing compounds targeting the ClpP. But we'll discuss two types of compounds, and then I will discuss the consequences of targeting ClpP. So ClpP is a serine protease, so of course you can inhibit it, but that turns out not to be a good strategy. Actually, no group has been successful at finding inhibitors that do work in cancer targeting ClpP. But what we have been doing, as well as other groups have been doing, is what we call activation of the ClpP. So what we mean by that is the following. If you look at the ClpP cylinder itself, so there are these hydrophobic pockets in purple, and these are the pockets where the cap docks. So this is a side view, this is a top view. And this would be the axial pore, the entrance pore for the ClpP. In the absence of the cap or in the absence of any other agonists, this pore is really blocked more or less, and only small peptides can go through. However, if you add a compound that open up that entrance pore, what can happen is that then a folded protein or something that has maybe an extended loop region can actually fall in to the ClpP chamber and get degraded unspecifically. So these compounds will prevent the cap from binding, but they also open up that entrance pore, and therefore they allow ClpP to degrade proteins unspecifically, and this eventually leads to cell death. So this is what we call is activation of the ClpP, so, or dysregulation of the ClpP. So I will be using this term activation of the ClpP. So I'll describe two different compounds that we've worked on. One are the acyl-depsy peptides, which is basically a cyclical type peptide, and the other one, which is based on an imipridone moiety that have this activating capacity. So the acyl-depsy peptides initially were discovered as antibiotics. They are natural products, actually, that are made by streptomyces. And eventually it was found that they actually target the ClpP to activate it. The ONCTO one is an interesting story, which I will mention how we stumbled onto it. So initially we were working on the ADAPs, and we were making many different analogs of the ADAPs. And the reason we worked on the ADAPs was from an antibiotic perspective. At the same time, when we were doing many different ADAP analog, and when I say many, actually we made around 50 to 60 analogs of these ADAPs. And this was done in collaboration with Rob Beatty's group. we were screening them against the bacterial ClIP-P, but we were also counter screening against the human ClIP-P. And to our surprise, several of the ADAPs that we made worked very well against the human ClIP-P. So this is what we call an RD. This is a quantitative number that we developed to give us an idea of how well these compounds activate the ClIP-P. So the closer the number is to 1, the better the compounds are. And several of these compounds worked very well against the human ClIP-P. So this was quite surprising for us because we were trying to counter screen against human, but they worked. And I'm showing you some of the structures of these compounds like ADAP28 and ADAP41. And I can show you that they do work indeed on a gel, for example. So you have ClIP-P, we can add the ClIP-X, and we add a model protein like casein. And ClIP-XP, the human ClIP-XP does not degrade casein very well under the conditions of this experiment. So when we added the ADAP at very low concentrations, we don't see much of a degradation. But then when we added it at a much higher concentration at 2 micromolar, we can see that the casein gets degraded. And we know now it's not ClIP-X dependent. It's actually ClIP-P dependent. So we can titrate the ADAP, and we get a KD of around 0.5 micromolar. So they are not too bad for an initial hit, 0.5 micromolar apparent binding constant to the human ClIP-P. However, the important experiment is the following, because initially when people in the lab started showing me this data, I thought this would not work on mammalian cells, because basically you have to think that these ADAPs, which are actually quite bulky molecules, they have to cross the plasma membrane, but they also have to cross the mitochondria to end up in the mitochondria and to end up hitting the ClIP-P. But what we found is that if you take something like HEK cells and you titrate the ADAPs, so this is wild type HEK cells, and we can see a normal titration curve. The IC50 is around 0.5 micromolar. But when we use HEK cells that are deleted of the ClIP-P, so there's no target for them, then they are not sensitive to the ADAP, telling us that we are on target hitting the ClIP-P in the mitochondria. And this was really the turning point for this assay, for our thinking that, yes, we can actually target specifically the ClIP-P in the mitochondria to activate the human ClIP-P and dysregulate it and causing cell death. We can see a direct correlation between the IC50 on cells and the binding constant that we measure on protein. This is because we have many different ADAPs. We have a nice correlation. Again, this tells you that we are on target. And we have IC50s within 0.5 micromolar for many cell lines that we tested, whether HEK, HeLa, Z2OS, etc. So we tested many different cell lines. And although the levels of the ClIP-P in these different cell lines are different, but we always get around 0.5 micromolar IC50. Now, the way that the ClIP-P is killing these cells is through apoptosis. Basically, we can show that by a tunnel assay. You can directly see the fragmentation and condensation of the DNA. And basically, this is a percent of cells with DNA strand breaks in wild type versus in the ClIP-P knockout. So you don't see these strand breaks because you don't have the target. The other thing that was interesting, we did check what's happening with different components in the cells, for example. So if you just concentrate on the slide here, this is the ADAP added to the wild type or the ClIP-P knockout cells. So you can see the levels of the ClIP-P are the same, plus or minus ADAP, but actually ClIP-X itself disappears when we add the compound. So the MCL1, which is an anti-apoptotic protein, also is downregulated. PUMA, which is pro-apoptotic, goes up. And we know that this is actually caspase dependent because we can see the activation of the caspase 9, which is the intrinsic caspase dependent pathway. It's cleaved and therefore it's activated, but the extrinsic one, which is the caspase 8, is not cleaved. So we know that it is a caspase dependent apoptotic pathway that's being triggered when we target the ClIP-P. I can show you these are 3D sim experiments, imaging of the mitochondria. So this is DMSO treatment, 24 hours and 72 hours. You can see very nicely the mitochondrial network. But then when we add the ADAP, we can see that the mitochondria is fragmenting, and we are basically eventually causing apoptosis. We also solved the crystal structure of the ADAP in the presence of the ClIP-P, and they sit where we thought they should sit, which is basically at these hydrophobic pockets shown here. And these are the hydrophobic pockets between two different subunits. And basically, that's how the ADAP allows the opening. There's through an allosteric pathway, it opens up the entrance pore and prevents at the same time the cap from binding, the ClIP-X from binding. This is the apo-human ClIP-P, and you can see that it is much narrower pore. This is a zoom in into that structure between the two subunits in the apo state. So one subunit is cyan, the other one is in purple. And then this is in the presence of the ADAP. You can see that the ADAP basically sits as a wedge between the two subunits. And this causes an allosteric effect that eventually opens up the entrance pore. So we continue to work on these ADAPs to try to further optimize them. But the best we are on cells is still in the micromolar range, which is not really great. So subsequently, we started working on another chemical scaffold, which we call the TR compounds. And these TR compounds look something like this. They are based on this compound called ONC201. So they have what's called here, this moiety is called an imipridone moiety. And this started as a collaboration with a company in North Carolina called Madeira Therapeutics. And this compound has an interesting story because basically, initially, it was thought to target a dopamine receptor, DRD2. So this is a dopamine receptor. And actually, the compound was already in clinical trials against multiple cancers, and there was around 15 clinical trials using this ONC201. And actually, it was even recently approved for glioblastomas. So it's being put in humans to treat glioblastomas. However, it turned out it does not target the dopamine receptor. It actually targets ClIP-P. And I'll show you that data. So we worked with this company, and we made many different analogs. I'll just show 5 of them. So basically, what we try to do is make changes to either the imipridone moiety itself or to the eastern or the western fragment. And we call these compounds TR compounds, so TR27, 57, 65, 107, 133, and so forth. So we tested these compounds against 60 different cell lines, and they work very well against all these different cell lines. So this is TR57 and 107 against these different cell lines. And they inhibit the growth of all these cell lines, and they even cause killing for a couple of these cell lines, like a colon cancer one and the breast cancer one. And when we measured the IC50s against MB231, so this is triple negative breast cancer cell line, what we have now are IC50s in the nanomolar range. So the units here are nanomolar. So it's really much, much better than our previous ADAP compounds. And we are basically between 10, 20, maybe 100 nanomolar. This is the original ONC201, which is in the micromolar range. If you knock out the ClIP-P, the compounds don't work, so we are on target. If we knock out the ClIP-X, the compounds still work. We also measured the KD on the protein level, and they are mimic what we see on the cell level. Basically, the KD, apparent KD, is also in the nanomolar range. So between 15 to, let's say, around 30 nanomolar. The ONC201 is in the micromolar range, 3.55 micromolar. So just to show you what the improvements that we made. So this is the imipridone moiety. What we found is that we can simplify this moiety further. Sorry, on the eastern end, what we can do is basically we had to add an alkyne or a nitrile group that helped in increasing the affinity of that compound. And in the middle, as I was mentioning, the imipridone moiety, we were able to simplify it to a pyrimidine type moiety or a pyrimidine dione with 2 oxygens here. And on the western end, we had to add something that's a bit electronegative, like a chlorine or a bromine. That helped a lot in enhancing the affinity of these compounds. So basically, we went from a micromolar again to a nanomolar. So Mark then solved the structure of these compounds with the ClIP-P. We have 5 structures with each of these compounds. And I'll show you just a zoom in into that structure. So basically, this is the overlay of the 5 compounds in the binding pocket. And you can see it fits very beautifully. So the imipridone is actually this flat surface. And then this is the eastern end, and this is the western end. And they fit into these pockets like very beautiful surface complementation. And that's why we are in the nanomolar range. So if I overlay, if I actually zoom in a little bit further, we can, sorry, I zoom out a little bit to show you the binding pocket. So again, this is the TR compound. And we think there's further space down here that we can maybe try to fill by extending this chain to try to further increase that affinity from nanomolar to picomolar. Just to show you the overlay between the ADAP, our micromolar binder. So the ADAP here is in purple, and the TR compound is in orange. And you can see that the TR compound is a much better fit to the binding pocket compared to the ADAP compound. The ADAP compound is bulkier, so it has this acyldecyl, the circular peptide up here. This part is very similar to the TR compound, but this part, the ADAP, does not fill this subpocket very well. And that's part of the problem with these ADAPs. So the consequences of ClpP activation. So initially, I'll discuss what we try to do on the cell level. So one thing that we knew is basically you're adding a compound, you're activating the protease. So I wanted to know, what is the protease cleaving? So the ClpP cleaves, and now it does not cleave processively anymore, but it just cleaves and leaves, right? So it's cleaving proteins. So we wanted to identify these new N-termini that are being generated when the ClpP cleaves a protein. And what we did is this high-chain experiment. We collaborated with Anne-Claude for these experiments, whereby we take, let's say, the MB231's wild type or knockout, and then we treat with compound or with the MSO for a certain amount of time, and then harvest the cells. Subsequently, we have to go through a process that allows us to identify this N-terminus. And this is the high-chain experiment. There's another experiment that's called TAILS, which has a similar idea. But basically, what you are doing is initially, you are blocking all three N-termini using by basically a formaldehyde. So there's a dimethylation that happens on all amino lysines and free N-termini. So basically, the lysines are blocked, the N-terminus is blocked with this dimethyl group. Subsequently, you trypsin digest, and then you are generating basically the N-terminus here that's further treated with a hydrophobic, very hydrophobic sequence. So this is in red. But this purple guy will not be tagged by this hydrophobic sequence. So then these peptides are removed on an HPLC column. And what is left is basically this purple N-terminus that's identified then subsequently by mass spec. So this is what we did. We did this high-chain experiment to identify all these new N-termini that are being generated upon treatment with these compounds. And therefore, you can do a lot of analysis. I'll just highlight a few things that we found. Basically, it was surprising. We found that proteins in the mitochondria do get cleaved, which is what we expected. But we also have proteins in the cytoplasm and the nucleus that are being cleaved. Now, why is that? The ClpP or the mitochondria maybe is opening up, and that's why the ClpP is getting out. That's something we are investigating very heavily currently. But what we see is that the proteins in the mitochondria, they get cleaved, but their levels go down. The proteins in the cytoplasm get cleaved, but their levels are stabilized. So this is one interesting feature. We don't completely understand it. We know what these proteins are. Of course, we identified them. And what we find, there's a lot of them involved in protein biosynthesis, maturation. But for this group, a lot of them are actually also molecular chaperones. So this was nice to see. Since we have the sequences of these proteins, we can ask the question of where the ClpP, the activated ClpP is cleaving. And what we see is that it is not cleaving at specific sequences. We know that ClpP is more or less promiscuous. It does not matter what sequence there is, but it mostly cleaves in random coil regions. So this is shown here. So most of the cleavage is happening in random coil regions, and this is what we would expect in a way, in retrospect. And I'm just showing you some of these proteins that we then verified that they are indeed being downregulated upon compound addition. So this is treating with TR107, the wild type versus the ClpP knockout. And the ClpX itself, which is I highlighted before, its levels goes down when we add with the compound. SDHA, which is complex 2, is part of the complex 2, it goes down. TFAM, which is the major transcription factor in the mitochondria, also goes down. TOFM, which is the equivalent, for example, of EFTU in the bacteria, also goes down. So we see, and some of these we verified biochemically. The other protein I'm going to highlight that goes down is this RNA polymerase, which is the mitochondrial RNA polymerase. Its levels also go down. And then finally, the chaperone, as I mentioned, this is HSP60, which is of interest to me, to us, I guess, people who work in the chaperone field. It's also targeted by the activated ClpP. Its levels go down. So however, I have to emphasize here whether not all of them we identify as a direct hit. There might be downstream effects. So, for example, TFAM we identified as a direct hit, but others we could not identify as a direct hit. Subsequently, we worked again with a group at the NIH to see the effect of these compounds on in an animal model. So this is a breast cancer xenograft mouse model, and treated with TR107, and measuring this is they use a certain regimen for treatment, and you measure the tumor volume in a control group, so you have a certain number. But then in treating these tumors with TR107, the volume is down. Basically, the compound is working. This is survival. So the group 1 is the control group, so they die earlier than the group 2 or 3, which is the compound treated group. Finally, they also measured a mitochondrial DNA copy number, and this is the vehicle control, and this is the group that was treated with TR107. Since we are mitochondria, we should really have a reduced mitochondrial DNA copy number, and this is what we see on average in a mouse model. So this is really gratifying. Of course, the treatment here was quite long, around 26 days. So we start to do better and try to further optimize, even though we are in the nanomolar range, or to have a better access to the cancer. So breast cancer turns out to be actually a very tough cancer model to work with in this context. So we might be looking at other cancer types. So on the protein level, I'm not going to go into too much detail of the biochemistry, but I'm going to summarize what we have found. And basically, on the protein level, what we found is the following. This is ClpP in the apostate, and this is the axial pore. Now, at the axial pore, there are these axial loops, which are shown here, and they are typically unstructured in the apostate. Basically, they are unstructured, and they have basically act as a barrier to entry into the ClpP chamber. And there's this ionic interaction network at the entrance pore of the ClpP. However, when you add the compound, the ADAP or the TR, as I mentioned before, they act as a wedge between separating the two subunits. And this results in the reordering of this interaction network. And we see that by X-ray crystallography and other biochemical means. And what happens is that now these axial pores are ordered because there's now interactions that are happening between the body of the ClpP and the axial loops that orders these axial loops and basically forces these axial loops away from the axial pore. And therefore, you're opening up that axial pore. So this is one feature of these compounds. They order the N-terminal loops. Another feature we also saw is that there's also ordering in the equatorial region. So this is how we think of the ClpP in the apostate. There's a lot of flexibility in the axial loops. There's a lot of flexibility in the equatorial region in yellow. But when we add the compounds, then the axial loops are structured, as well as the equatorial region is further structured, is further stabilized. And you think of it basically that when you add the compound, you open up the entrance of the ClpP, but basically you tighten the belt or you shrink the equatorial region. So ClpP now is able to eat more, but it remains thinner, becomes thinner by eating more. So this is a very interesting feature, and there's a lot of allostery in this cylinder that has to happen. So with this, I'd like to conclude by hopefully convincing you that this regulation of the mitochondrial ClpP is a novel approach to develop new anti-cancer agents, but also this leads to very interesting biochemistry, and biochemistry is really what we are, biochemistry and structural biology is really what we are very much interested in pursuing, but combining it with this medicinal chemistry approaches and chemical biology approaches is something that has led us into new paths. And finally, I'd like to acknowledge my group again, especially here is Elisa has done a lot of the biochemistry, as well as Mariam here in the back. The structural biology was done by Mark, who's unfortunately not in this figure, but the cell biology was done by Keith. And now we have Mazen and and Funning has done a lot of the, Funning, I should not forget him, he's done a lot of the medicinal chemistry. So a lot of the compound synthesis. So with this, I will stop and take any questions.
Awesome. Thanks, Walid. That was really exciting and interesting work. Do we have any questions? Again, either feel free to paste them in the chat or raise your hand.
Oops, I can still share.
Yeah, sure. Oh, Fabian, I will unmute you. Go ahead.
All right, awesome. Thank you. Yeah, thanks, Walid. That was a really great talk. I got a ton of questions, but I'll just ask one out of curiosity. So you have all these ADAP and tR variants, or like different molecules to look at the two ClpP proteins. Do you know if in parallel people are looking at the variants that don't bind that good to the human ClpPs as a potential antibiotic? Because if the way you explained it, it sounds like it should kill bacteria also pretty efficiently.
Yeah, actually, so we're doing antibiotics as well. This is another talk that I give. I have an anti-cancer talk and I have an antibiotic talk. So so we are developing the ClpPs, so anti-ClpP as antibiotics as well, and we have a whole collaboration again with Rob Beatty that we're doing that. Interestingly, so the ADAPs work very well, much better against the bacterial ClpPs than the human, even though we're trying to optimize them. We try to optimize them further for the human, but we've been more successful with bacteria. So bacteria ADAPs are fantastic. They are really like 0.0001, they're MICs. But the TR compounds, which work very well against the bacteria, against the human, I mean, they don't work well against the bacteria. And we know why. So the binding pocket is different between bacteria and the human. So it turns out that in the human, there's the tryptophan that blocks the binding of some of these compounds. And that's basically the main difference. One of the differences between the two.
Okay,
yeah.
Really awesome. No, thank you so much.
I can give another talk, another hour talk on the antibiotic angle. Yeah.
I'll look forward to it. Thank you so much, Walid.
Sure.
Awesome. Suanne, I'll unmute you.
Oh, hi.
Hey,
Hi, Walid.
Suanne, how are you?
Good. Just beautiful work. I have a lot of questions too, but I'm going to try and limit myself as well, just to one.
Sure.
So I guess I'm really curious because you're missing the unfolding function of the ClpX, right? So in these experiments, like, do you find that there is more kind of cleavage at the N and C-terminal regions of proteins, like resulting in kind of like partially degraded proteins that might have new functions?
Yeah, so basically what is happening is that initially I mentioned, so when you have ClpXP, it's very processive, so you don't see intermediates. But when you have ClpP that is activated by compounds, then you can see intermediates. Even on a gel, you see intermediates. And as I mentioned, most of the cutting seems to be happening in coiled regions, random coiled regions. So any floppy parts of the protein are being attacked by the ClpP or have access to the ClpP catalytic chamber.
Yeah, I'm just curious, like if those intermediates like could clog the ClpP or have new kind of like functions that are kind of contributing to the phenotypes you're seeing.
I mean, officially, I don't know that, but I mean, what we see is in some cell lines, as I showed, we eventually lead to apoptosis. So basically the cells completely die. Or in other cell lines, which seem to be more resistant, we have cell cycle arrest. So basically they go into senescence. So these are the two outcomes that we've seen. Whether that's because of a specific protein that's new protein in a way that that we are generating. I cannot say that, but I think it's something to do. We are really messing up with the mitochondria eventually.
Okay, thanks.
Okay, Siavash, go ahead.
Hey, Siavash.
Hello. Lovely talk, Walid, as always. I wanted to get your thoughts on why is it that the ClpP complex appears to be in the compressed/compact form when bound to your compounds, the activators?
No, it is in the extended state when it's bound to the activators. Yeah, not in the compressed.
Oh, is it?
No, no.
Okay,
We had hoped to see it in the other states, but crystallographically more. So in the bacterial ones, we've seen some in a sort of a compacted state, but in the human, we've never seen. Let me see. In the human, some of the structures are in the compacted state. Actually, I take that back. Some of the structures that we have solved are in the compacted state. I think, again, it's an issue of dynamics. I think what's happening, you have basically, it's always switching between the two. And, but then when you have the compound, you're basically populating one state more so than the others. We've been doing cryo-EM on the ClpP these days a lot. And the human one, we never seen it in an in a compact state. It's always been in a extended state, plus or minus a compound. I know you've done the bacterial one, and it turns out to be there's some pH dependence to that. So that's very interesting because that's something maybe we would look at, because as you know, in the mitochondria, your pH is like a bit more, 7.8 to 8 in the mitochondrial matrix. So you're more basic. So that there might be something to look at there as well.
Perfect.
Yeah, very
Thank
good
you.
question.
Okay, Gabe, I'll unmute you. Oh, there you go.
That's on me. Hi, Walid. I apologize for muting myself after Matt did the work. Yeah, great presentation. I'm really curious about whether you've looked at the mechanisms behind ClpX, kind of like stark degradation. I'm assuming it's degradation, like removal of protein, basically, when you.
Yeah, why ClpX disappears?
Yeah.
Right. So this is something we've been looking at, and so if you use purified proteins. So let's say we just purify ClpP, have a ClpX, add the drug. We don't see ClpX getting degraded in a purified context. So at least in a purified system, the activated ClpP cannot degrade ClpX. So something else is going on. So one group proposed that it is lone, which is the other protease in the matrix that does it. However, we checked for that. Actually, our collaborator checked for that, and he says it's not lone either. So we're not sure what's happening with the ClpX, and it's really very quick, and it disappears completely.
Yeah. So from my experience with working with protein like one particular protein complex, when you remove one subunit, all the other subunits, even though they're not usually prone to degradation through the proteasome, they get proteasomally degraded. So that might be something if ClpX is constitutively and it's very stable when it's interacting with ClpP. When it's not there, then maybe there are some lysine residues or some other modifications that could happen that lead to its degradation. So
yeah, so basically, we checked for that. But so basically, if we knock out ClpP, if you knock it out, ClpX is still there. It doesn't matter. It's only when we activate the ClpP, ClpX disappears. So it's a bit puzzling for us, and we're very interested to figure this out as well. So that's something we're also looking at.
Good stuff. Thank you.
But good question, yeah.
Awesome. I think, seeing as there's no more questions, I think we'll wrap it up there. I want to thank both of our speakers for giving really great talks today. That was some really great science. And before we go, I just want to plug our next seminar next month. So it'll be on Tuesday, February 6th at the same time. We have another set of really amazing speakers, and we hope you'll join us. So thank you everyone for coming, and I hope you have a good day.
Thank you very much. Bye-bye.